Welcome, everybody, to our second RIDE seminar. My name is Kevin Lang. I'm a professor in the VBS department. I've been helping get this seminar series on the road here started. So today we have a really exciting invited speaker. And so I'm going to hand it off to Dr. Peter Larson to introduce our speaker. So Peter, take it away.
Thanks, Kevin. Thanks everyone for coming today. It's a great crowd. We are so excited to have Dr. Belsare here with us today. We're trying to get him here. There's so many common interests. We've been talking for years now, and so Ani is here to share some of his research with us. But we're thrilled to be hosting him through the Minnesota Center for Prion Research and Outreach, or MINPRO. Dr. Belsare is a disease ecologist with a background in veterinary medicine, pathogen modeling, and conservation research. He obtained his veterinary degree from the Bombay Veterinary College, Mumbai, India, and his PhD from the University of Missouri. Currently, he is an assistant professor of disease ecology in the College of Forestry, Wildlife, and Environment at Auburn University. A major focus of Ani's research is on host pathogen systems that have public health or conservation implications. He uses an interdisciplinary approach that incorporates ecologic, epidemiologic, and model-based investigations to understand how pathogens spread through, persist in, and impact host populations. One of the main objectives of Ani's research is to translate insights gained from an analytical approach into actionable outcomes for managers and policymakers. And Ani was one of the very first scientists to reach out to us back in 2019 when we started our research effort on chronic wasting disease. He has so many fantastic ideas. The technologies that we've been developing out of MINPRO, he has clear applications for in the real world and putting it all together into his modeling perspective. So with that, I'll hand it off to Ani. Let's welcome him for the seminar today.
Thank you so much, Peter, for the introduction. And I have to say this, I am super excited. I'm super excited to be here. Really, because as Peter said, we have been communicating for a while. And this has always been my question, and I have said that oftentimes to Peter, like, why don't I see agencies lining outside your lab saying, we want this technology? And, this is the first time I was able to meet with the team of MINPRO, and we just couldn't stop talking about their work and CWD and the potential, applications and all of that. So I am going to talk about the applied epidemiological research that can be done using an agent-based modeling approach. One thing I'm not going to do is I will not talk about chronic wasting disease today, though that has been a major focus of my work for the last decade or so. But because yesterday I have talked about CWD for all day, and most people here, I know, probably have had a lot to hear from me about CWD. But so what I have decided is I'm going to talk about two other projects that I have been working on for a number of years, and at present we are trying to springboard using the work that we have done on those two disease systems, namely SARS-CoV-2 in white-tailed deer, which builds upon the chronic wasting disease model, and leptospirosis, which is a neglected disease, and it's really not limited to the developing world. It's a notifiable disease in the US as well. So these are the two disease systems I will talk about. And so I do have to start with disease ecology 101. And what that means is the epidemiological triad or the disease ecology triangle, whatever you want to call it. What this tells us, this kind of gives us an idea about the complexity of these systems. And there is a real danger of not acknowledging or not incorporating these complexities into our understanding. And it oftentimes has resulted into scenarios where the remedies that we came up with for dealing with these complex disease systems were actually dangerous than the maladies themselves. And so that is a real danger. And so what I want to say here is we know infectious diseases, right? They have, for any infectious disease, the pathogen has to interact with the susceptible host for the disease to be transmitted, right? And it is not as simple as just a host meeting or getting in contact with the pathogen. Most of the pathogens that we know, most of the pathogens that humans are infected with are generalists, host generalists. So they can affect more than one host species. And the interaction between these host species is a real challenge because they affect the way the disease kind of, persists or spreads into the community of the hosts. There is also this thing which is not always talked about, not always acknowledged, but it's well known, which is the host heterogeneity. So I always bring up George Orwell's phrase about all animals are equal, but some are more equal. It's the exact same thing. All hosts, they're not equal. Even within a species, like you have age and sex and all those differences, which result into patterns that we need to understand if we have to effectively mitigate the threats posed by infectious diseases. There is also the issue of co-infection, which we just talked this morning about. Like, again, it's a very simplistic understanding if we just assume that a host is infected by just one pathogen at a time. It's never the case. There are multiple pathogens, multiple types of pathogens within a host which interact with each other and can result into a completely different outcome. And there are phenomenal studies that are out there about this, but all of this needs to be taken into consideration if we really have to kind of mitigate the threats posed by some of these infectious diseases. And there have been a lot in the recent times. There are vectors, the vector biology, the range expansion, climate change, and so on, multiple interacting components in this system that interact with each other and which gives a myriad of outcomes. And that is the real challenge of managing diseases. And I am going to limit this discussion to diseases of wildlife or of free-ranging animals. They include the synanthropic animals that are around humans, and they all pose kind of the similar challenges. The hosts and their pathogens move around freely, right? They are difficult to access. If it's a wildlife thing, you have to access them, capturing, trapping, surveillance, everything is a big problem. Added to that, we really don't have any tools. Like there are exceptions, like rabies, raccoon rabies in the eastern US, you have oral weight vaccine. And there has been quite a successful program for the last 3 decades of limiting the expansion of raccoon variant of rabies westwards. Though last year was a bit of a issue because they found the raccoon variant much farther west than they kind of were hoping to limit it. Anyways, my point being the tools are limited. Okay. What that means is if we do not incorporate this understanding of uncertainty, of complexity in our response oftentimes will be quixotic. Like there will be imaginary giants that we will be fighting out there with all good intentions, yet it's not going to help us because we fail to acknowledge all these interactions in there. What do we do? What do we do with such complex systems? And it has been always recommended for any complex system to use a triangulation approach. What that means is just multiple current and relevant methodologies, gather information about the system using all these relevant methods and put them together and then better understand the dynamics of that system. And that's going to be the focus of my talk today, wherein how can we better understand the dynamics, let's call it like the pathogen dynamics or transmission dynamics in these complex systems. Also, can we use these model-based tools to compare and contrast interventions? How effective are these interventions? So just to give you an example, oftentimes, let's say for a disease like leptospirosis, it is a neglected disease for a reason. It affects mostly poor people, right? So the priority for dealing with leptospirosis is not very high out there. So if you notice, much of the recommendations talk about rodent control or control of other hosts like free roaming dogs, or educate people to protect themselves from environmental sources of like leptospira. Now the issue is this is all good on paper, but if you really go to an urban slum, this doesn't really matter. Rodent control is impossible. no place on this earth there has been successful rodent control. I believe there is just one region in Canada where they have done that. But otherwise, rodent control doesn't happen. Dog control, free roaming dogs, like is a big issue in the developing world. And so these recommendations are often out there, recommended by people who do not have mud on their boots, and they are very difficult to implement. So that is one reason why, or one thing I always complain about anytime there is a leptospirosis outbreak in India, Africa, or wherever on the ProMed list, there is some expert who comments, oh, they should use doxycycline as a prophylaxis before the monsoon season. And I just wonder, like, man, do you know how many people live in slums there? And like, antibiotic resistance is real. Doxycycline does not have any special power to evade that antibiotic resistance. Is that really a solution? Then the question comes like, then what is the solution, right? And this is where I kind of jump into how modeling can help. And really speaking, what I want to emphasize is it's not only integration of data and better understand the dynamics of disease and use it as a tool, because you cannot really go out in the field and say, I will go and kill 5,000 white-tailed deer and see its effect on the spread of chronic wasting disease. These are the things you cannot really do in the real world. But the model as a tool gives you the ability to do a what if scenario analysis. And that can then come back to like an actionable outcome for disease management. So those are the main things. I just sum it up by saying it is like finding the Achilles heel of this complex system. If we are able to find that, we will always have uncertainties about the complex systems. We will never know all that is needed to know, but we can still kind of manage this wicked problem if we find that Achilles heel, and that is the purpose of modeling here. The reason I am excited to be here, I was in Minnesota in 2018 for the leptospirosis work, but this paper published in 1997, this is the sole reason why I got into disease modeling. I read this paper in 2008 during my PhD program at University of Missouri. I had no idea what modeling was before that. And if you had asked me then, I would have said like, oh, it is just something people do on computers with no real world value. But this paper, so Anthony Starfield was a professor here in University of Minnesota. He's still an emeritus professor. I have never met him, but if you know him, please tell him he has influenced my career. I am a modeler just because of this paper. And it's it's a short essay. It's not like a big thing, but there are several interesting things here. He talks about like this is way back in 1997, and this is kind of intended for wildlife managers, wildlife agencies. All of those points are applicable to wildlife disease management. And the essence here, I think, is using models as a problem-solving tool, right? You never, so we again discussed this yesterday, I will be repeating some of that stuff, but you don't model despite uncertainties. You model because of uncertainties. That is the thing. The uncertainties don't go away, but you quantify some of that part and then are able to make better decisions. So Starfield says, use them as a problem-solving tool in all of this uncertain kind of, complex systems. And that is one thing that remained with me. I kind of summarize it during the other workshops as AIDS decision-making, which is like, you can assess threat realistically in a real world system with diseases or a disease that is spreading in the population of your interest. You can assess threats realistically using a model-based approach. You can better understand the transmission dynamics and therefore will be able to make better decisions. And we will see some of that in the next two case studies. Design, compare and contrast interventions, like what works, what might not work, which one is better. Even though you cannot test that in the real world, or if you can test it in the real world, it might take a long time. Like some intervention for chronic wasting disease, let's try it for 10 years. And then after 10 years, let's come together and see whether it worked well. If it hasn't worked, the disease is already all across the US in all counties and all states. So that is not a good idea. So we have a quantitative tool to evaluate, compare, and contrast all of this. So again, this is a quote from the paper. The question is not whether to model. It is how to model usefully and efficiently. That is the crux of the work that I do. Okay. Why agent-based models? There are so many other approaches to modeling. I am just going to summarize this pretty quickly, but I think the main reason I recommend agent-based models is because in this kind of modeling, we use data from individual animals, right? And the actual process of disease transmission, the very fundamental process, is as we saw, a susceptible host has to get in contact with the pathogen, which might come from another infected host or an infectious host or from the environment. But the process is very individual based process, right? It actually happens at individual levels. And that is what we do in agent-based modeling. We build these models. They are comprised of agents and they can have multiple types of agents which interact locally. And we see the pattern globally. The agents interact in time and in space, and we can project these interactions over the desired number of years or months or weeks. And then that pattern, that emerging pattern, is what gives us an idea about the real world transmission, the real world issues with those kind of disease systems. Very quickly, so what you see here is a population of, let's say, white-tailed deer, and each of these deer is represented in the model as an agent, as an individual, or in the NetLogo terminology, they call them turtles, right? So each of this agent has a set of attributes, real world attributes like age and sex and whatnot. So they can be many, like immune status. Their behavior can be incorporated. That is a big plus point compared to the other methods of disease modeling, like compartmental modeling or some kind of statistical modeling. So we can get all these real world processes in the model. They are spatially explicit. And to really sum it up, this is what gives you the ability to design and implement virtual experiments in this in silico world that you have created. You are the god of this world. You can go in and interrogate this system in any ways you want. And I will show some of that here, just a brief kind of, overview of what I find most exciting. So the first case study, which builds upon the chronic wasting disease model, is the SARS-CoV-2 model in white-tailed deer. It is important for multiple reasons. It kind of is a unique problem. There are many other species who are susceptible to SARS-CoV-2, and they also have the ability to sustain the transmission. But white-tailed deer, at least in North America, play a very special role. You have 30 million white-tailed deer across the rural-urban gradients, and there is a very significant human-deer kind of, interaction scope. There is hunting, there is recreation, deer come to the backyards, to the gardens, and all of that. Having said that, there are multiple studies which show that there is ongoing transmission, or there is spillover from humans into white-tailed deer populations, ongoing transmission into white-tailed deer populations, and there have been a few studies recently which show spill back from white-tailed deer into humans. Some of those show that the variants which went extinct into the human populations sustained in the white-tailed deer populations and then spilled back into the human population. So it poses a very intriguing problem. We don't know how the pandemic is going to shape out with a novel wildlife reservoir like white-tailed deer or some others, but we still need to know the patterns, the transmission dynamics to make better decisions. And this is where we decided to adopt the chronic wasting disease model to a SARS-CoV-2 model. And I'm going to present some of the preliminary results here. Before I do that, I just want to point out some of the terminology. So if you read the literature, oftentimes you will notice terms like spill back and spill over used interchangeably, or they kind of mean something different than other papers have used the terms, So what I want to point out, this is based on the framework, a conceptual framework that Fenton and Pedersen proposed in 2005. A very interesting paper I always use in my disease ecology classes. But what we need to understand is there were multiple spillover events. right? And so the spillover event kind of are in the lower quadrant here, where the between species transmission is low, but the within species transmission is absolutely not there. So, for example, the pangolin got infected by the virus from a bat, possibly which was in the cage above it, but it got infected. It had the virus, but it had no ability to pass it to other pangolins. There was another spillover event into some unknown species, and there was another spillover event into the humans. Right? These were one-off events. But when it came to the human population, there was ongoing within species, within humans transmission, and that is what they call an emerging infectious disease. The point I want to make is the dynamics in a multi-host system is very important to keep in mind. Everything is not a one-off spillover. Not all spillovers will end into like a fade out. Some spillovers will have ongoing transmission in the affected species. And more importantly, now we are in a position which is neither spillover nor an emerging disease. It is a true multi-host pathogen. So we have multiple animal species that have variants of SARS-CoV-2 circulating in them, and they can jump between the species and humans or other animals anytime. And that creates an entirely different problem. So coming back to SARS-CoV-2, what we really want to understand using some sort of an analytical approach is what is the dynamics in this novel wildlife reservoir, which is everywhere, right? And are there ways that we can make intelligent decisions about preventing human infections, recurrent human infections, or white-tailed deer are not affected by SARS-CoV-2. So it's not a conservation issue at this point of time. They get infected, they don't show any signs. 6-7 days, they're out of it. They generate antibodies, which about 6 months you can detect them, and then the antibodies are gone. So it's not a wildlife conservation issue, but can it be a public health issue? And if it is, are there ways of understanding it beforehand rather than wait for another 5 years and then, so that's the case here. You have circulating SARS-CoV-2 in the human population. Once in a while, there is a spillover, and I'm calling it a spillover. Some papers call this a spillback because they assume that animals are the source. Spillover happens into humans, and then human to animal is a spillback. Doesn't matter. It is just the interspecies jump that has been shown to regularly happen, at least in North America. Then you have ongoing infection in the white-tailed deer population, right? Deer-to-deer transmission. They're very efficient. So it does spread in regional deer populations. The real worry is of this variant of concern, because the longer this kind of a pathogen circulates in a host population, greater are the chances of adaptation, evolution, or whatever. And will that result into a variant which probably can escape the immunity human population has developed? Will that have the ability to kill more people, and so on and so forth? There was a posting on ProMed yesterday from Syria. I don't think it has to do with any animal reservoirs, but they are kind of considering whether the new variant there, I think it's J1 or whatever, there is a kind of sentiment that it's killing more people. SARS-CoV-2 again, but it's not all verified, it's just a report. But that is the real worry, variant of concerns, right? And then spill back or spill over into human populations. So the model that I am going to talk about is limited to deer transmission, and how best can we learn from the dynamics to make decisions about surveillance, to make decisions about the possibility of emergence of VOCs, and to prevent spillback. So again, these are some of the recent papers where they talk about may serve as reservoir for extinct variants of SARS-CoV-2, accelerated evolution of SARS-CoV-2 in white-tailed deer, something of concern, and multiple spillovers from humans to deer, and then onward transmission. So this has been, people have been looking at it, and one of the things I think, that I found very interesting is we already had a framework to simulate transmission of a disease in white-tailed deer population, right? This framework has analytical flexibility. It is very very portable and customizable in the sense that it doesn't have to be chronic wasting disease. It doesn't have to be white-tailed deer. If you are interested in some other host species, you can just recalibrate it, reparameterize it, and run it for that host pathogen system. So the first task was, can we convert this into a SARS-CoV-2 model? And what I want to highlight here, what I want to really emphasize is deer, their social structure is peculiar. Right? They are not entirely social animals. Like they have a kind of a matrilineal system, but it's seasonal dynamics. So the doe with her newly born fawns or fawns of the same year, they form a very compact doe social group, right? Males are associated loosely, but they have like a buck group. Males and females do not interact except during the rutting season or the breeding season, and so on and so forth. So all of that, that dynamics of the behavior and the social grouping is incorporated in the model as a contact matrix. And just to kind of bring you back to like the very basic process of disease transmission from a modeling point of view, it is a probability of contact between an infectious individual and the susceptible individual, and the probability that transmission occurs given such contact, right? So this takes care of the probability of contact. And from some studies, we know the probability of transmission given an infectious contact has occurred. So we have used that in the model for the chronic wasting disease. And as I said, these are very customizable, portable models. We already had used them for different states. We have used it for Norway and reindeer. And so I was confident we will be able to bring in SARS-CoV-2 in white-tailed deer. Just important kind of points about these two disease systems. So misfolded prion, so not really a pathogen, but acts like a pathogen. And then you have a virus with the COVID or SARS-CoV-2 thing. There is no adaptive immunity. I think this is a very important distinction. Most diseases we know, viral or bacterial, there is an adaptive response, which we often measure, like the antibodies or the ELISA tests or seroprevalence is where we measure, we have some way, some metric of understanding the exposure patterns. Not so with chronic wasting disease. And CWD, as the name says, has a very prolonged course of disease, invariably fatal. COVID, they are not really affected, right? And then short course, no clinical signs and all of that. So very distinct disease systems. And what I essentially did for the COVID model is change the time step of the chronic wasting disease model, which was 1 month. So the model advanced in time steps of months. Because it was a chronic disease, we have long durations, it didn't matter. But for COVID, I converted it to a daily time step. So things are now happening daily in the model. Furthermore, we added adaptive immunity. So an infected deer in the COVID model generates antibodies, and then those antibodies wane over a period of time. So those are two important distinctions we had in there. And, the first output that you see there is the trajectory of COVID in the regional deer population. Keep in mind, these populations are not hypothetical or like theoretical. They actually represent real world populations. They are built using data from wildlife agencies. So this is Montcalm County, Michigan, that you see. The deer population, the deer numbers, the age structure, the composition, all matches from what we know from the real world Montcalm County data, right? And if I introduce SARS-CoV-2 in the white-tailed deer population of Montcalm County, you would see a trajectory which spans, at least in the graph down there, it spans about 4 years. So a single introduction can result into a long outbreak into the white-tailed deer population, but that's not all. On further investigations, before I go there, this is like the distinction. This is chronic wasting disease. X-axis is number of years. That is COVID in deers, and you just have days on there. And you can see the outbreak sizes and all of that. Here, notice that the prevalence remains very low, under 1% for the first 10, 15 years, right? That's not the case over there. And further investigation, and I think this is something very, very intriguing. Okay, I'll just spend a few minutes on this slide. But what you see here on the months on this y-axis, they are actually the spillover event. So a spillover event happening in August results into a very short outbreak in the regional deer population, right? The outbreak starts here somewhere around August and is done before the next breeding season, like parturition season, right? It's a very short outbreak in the local population. Not so if the introduction, the spillover happens in February or March or April. A single spillover results into a multi-year outbreak, right? This is something interesting, and I'll explain why. Because we take into consideration the deer behavioral patterns, the seasonal dynamics of how they interact, what's the social structure, how does it change. And it is mostly because of that behavioral pattern that you see different resultant outbreak trajectories just based on when the spillover happened. One potential application of this kind of a tool, we are trying to work on that, is combining this with wastewater surveillance data. So CDC does wastewater surveillance across US, and you can actually go in and look at that every month. So you have an idea of the virus circulation in human population, right? And that is one indicator of the probability of spillover into the regional deer populations. It also helps us design better surveillance because, let's say, if the harvest season is December, then that is when most of the humans, the hunters, are going to come in close contact with the deer they harvest or they kind of interact with, right? Does December present a high probability of infection or high probability of virus circulating in white-tailed deer in the local population? Possibly. Sometimes it is correct, sometimes it is not so. So can we predict these kind of patterns in regional deer populations? That is one thing we want to look at. But more importantly, as I said, we have included adaptive immunity. So we can also look at the antibody prevalence in the deer population, cut to the harvested deer, if along with the other testing, if the agency can look at seroprevalence of antibodies against SARS-CoV-2 in the regional populations, can that help us understand what is happening in the white-tailed deer population? Also added to that, yeah, seroprevalence is one thing, and this is a point I keep making, but the force of infection is something that the agent-based model helps us kind of derive or estimate, because as I said, you can go into the population and count everything. The force of infection here counts each and every infection happening every day. And then that gives you this metric, which is a standalone metric for the probability of infection in a susceptible deer in the population. Also think about the interaction between antibodies, so multiple spillover events. First spillover events results into rapid spread and buildup of antibodies in the white-tailed deer population. The second spillover event might not spread equally because now there are locally deer that have antibodies. That is not the case with chronic wasting disease. So you see really distinct patterns, and the hope is that can help us design better surveillance strategies, right? Not just more sampling, but kind of a better understanding of what's happening in the regional deer populations to have locally relevant strategies. Yeah, this is how we do the force of infection. We can actually count or design or kind of come with a who infected whom matrix for the given population. I am working with a evolutionary biologist, and we are trying to use this framework to come up with a probability of emergence of variants, not necessarily of concern, but do certain conditions result into a higher probability of emergence compared to other set of conditions? That is what we are trying to do right now. With that, I think I will just cut to the second case study here, which is the leptospirosis. This is, as I said, is a neglected disease. It's not limited to the developing world. 50,000 people die every year with this, and they keep on dying. Urban slums are increasing at a rapid pace. And so this disease will remain a cause of concern for a long time. Yet, because it is neglected, because of the catch-22 aspect of it, we keep on advocating the same things without actually, evaluating whether it has worked. So this is this is from Chile, and this study was done again with a faculty at University of Minnesota. And I was here in 2018, and it was an amazing time. We kind of collaborated and worked on this model. But this is based on the study Dr. Munoz Zanzi did in Chile. So the model is based or parameterized on the data from her field study, which represents urban slums in Chile. It's applicable to a lot of places too. So 1 million cases per year, more than 15,000 human deaths globally, underreported. So the real numbers might be a lot more. There are no control measures, as I said, like there is no human vaccine, no effective, safe human vaccine as of yet. So really speaking, the only thing you can do is address the infrastructure deficiencies. That is not going to happen, right? With more and more urban slums coming into and flooding and climate change and all of that, that is not going to happen. So we end up with recommendations like rodent control, dog control, free-ranging animal control, better protective measures for people living in slums and all of that. The compliance for all of this is really low, and so nothing really changes. So just to understand the complexity of this system, you have a system where you have multiple host species, and multiple serovars. So there is a lot more complexity than just one bacteria and one host species, okay? So you have the rodents thought of as the main reservoirs, and this is the sero-R, which is the rodent adapted sero-R, okay? Then you have dogs in this system, free roaming dogs with their own sero-R. Let's call it sero-D for now. So you have two different kind of sero-Rs, two different host adapted species. Humans always get the infection from the environment. The leptospires are excreted by the infected animal in their urine mostly, and they remain viable in the environment, and then humans get it, occasionally, frequently, whatever. The real confusion, the real complexity is here. So sero-R, which is the rodent adopted strain, okay, a rodent will remain persistently infected and will keep on shedding the leptospires intermittently. Right? This is the first part. But if a rodent is infected with sero-D, it can be infected, right? It will develop a lifelong immunity for sero-D after it recovers in a few months, right? There is a different response. Similarly, a dog infected with sero-R will have a lifelong immunity for sero-R after recovery. Not so with the sero-D, it will have a 1-year immunity after recovery, but then it is susceptible. So a lot of complex kind of interactions happening, and this is exactly what we built into the model to look at what patterns emerge and can it help our designing of the management strategies for this one. Okay. Yeah, so this is the model interface, and as you can see, just like the other model, there are a lot of sliders on there and a lot of plots. The reason for this is we want to make the models accessible to non-modelers, and that they can use it to design locally relevant strategy. It shouldn't be just the modeler doing his or her work in a room and sending out the results. So we are far from that, but that is our goal. Okay, I just want to highlight how we can use this kind of a model-based tool to evaluate or develop or test strategies that cannot be tested in the real world. So I just have a couple of 3 examples here. Rodent control, right? Let's assume that we are able to do rodent control in one such setting. The baseline is status quo, so no rodent control, right? Then you have a rodent control strategy which results into 25% reduction in the rodent population throughout the year. It's a very simplistic scenario, just to have a better feel of what's happening in there. And the RC50 is a strategy where we are able to suppress the rodent population to 50% of its original size. So you have 3 different scenarios. If you look at the plots, what you see is this A is sero-R, that is rodent adapted cases in rodents. The baseline is there, and green is 25, blue is 50. You see a definite decrease in the circulation of rodent sero-Rs in rodents if you control the rodent population. This says this is density dependent. The lower the density of rodents, the lower the circulation of the pathogen, right? Not so really with zero D cases in dogs. There is not much improvement there. So rodent control doesn't really have any effect on the zero D cases, the dog adapted zero R in dogs. The zero R cases in dogs do go down, right? The rodent adapted overall, it has a good effect. What is really important and what I want you to focus on is all of this should be connected to the human risk, the zoonotic risk, right? So for the baseline scenario, we had about 3,000 cases in rodents, but with the blue scenario, which is 50% reduction, we had significantly less number of cases in rodents, like 600 compared to 3,000. Similarly, the zero D cases in rodents, a dog adapted zero R, they did come down by 50%. But more important is there was 20% reduction in human outbreak probability. The possibility that a human will get infected in this kind of a control scenario came down by 20% with the first one and 40% by the second one. Now, these are not values that are predicting what will happen in the field, but these are important to compare with other interventions. Namely, let's look at so this is the density dependent part. Let's look at dog control. What if we go to the same slums and implement strategies wherein dogs are no more free roaming? So if we are able to bring down the free roaming dogs by 25% and 50%, how does that affect? So there is no impact on zero R cases in rodents, but there is a definite decline in zero D cases in dogs, right? And we can just look at how does that impact the human cases. So it's similar impact. Human cases go down by 20% and 30% in these two scenarios. Doesn't really mean much until you look at dog vaccination. Now this becomes more complex because the vaccine can be a monovalent vaccine which just protects dogs against zero D. And then you have a bivalent vaccine which protects dogs against both zero D and zero R. The best possible outcome you will see here, if you have a high coverage of bivalent vaccine, the cases literally go down in dogs, in rodents. It does not affect zero R circulation in rodents, right? I know this is all complex, but I've summarized it later on just to give you an idea how we can better understand all of this complex dynamics. So just focus on this one. You can get 50% reduction in human cases if you use a bivalent vaccine in dogs and have a high vaccination coverage. So that is great. But just keep in mind what that means is a low vaccination coverage in dogs or a vaccination in dogs with just the dog zero R doesn't really make much difference. It also tells us we can actually mix and match. We can design strategies of differential dog and rodent control and vaccination based on local conditions and find out which set of strategies make more sense. So in that way, as Starfield said, this is a tool that can be used in a locally relevant context, knowing what kind of hosts and what kind of host zeroars we have, and then design a locally relevant strategy rather than just saying, hey, let's control rodents, let's control dogs, let's vaccinate dogs, and so on. This also helped us better understand the dynamics in such a complex system. So I just want to highlight what you see here is rodents were actually able to sustain dog zero R, which is this, even when dogs did not sustain it. So this is something very important. If you stare at it long enough, you will realize like only 84 iterations had the persistence of zero D in this scenario, but actually, 5 more iterations, the zero D persisted in rodents, but it was not there in dogs. This has very important implications because it means the simplistic understanding, if you control one zero R in one host species, it's gone. It might not be the case, right? Also, notice that the contribution of leptospires to the environment, right? That is the main important part. That is the zoonotic kind of interface. How much leptospires are in the urine of the infected host and how many of those leptospires actually end up in the environment. It tells us that rodents play a very important role in the environmental contamination. right? Even though dogs put out a higher volume of urine, right? Yet it is the rodents which actually are the main threat or main contributors to the leptospires in the environment. And this is the realistic population. The number of rodents and number of dogs is based on real world data. It's not like we made up and this is like, what you see. Finally, I just want to sum it up. So what we found, and this paper has been referenced in the recent ACVIM guidelines on leptospirosis control. So that was encouraging. But rodents were the main contributors for both serovars. Rodents independently sustain the dog serovar in some cases. There is a density dependent transmission of the leptospira in the rodent populations, at least for the urban slum populations, because the densities are very high there. And dogs played a very important role in distributing the rodent adapted strain in the landscape. This is very important. So even though dogs were not the main contributors, if they got infected with the rodent serovar, they dispersed it in the landscape, kind of making it more available to the human host. So I just want to sum up, I guess I am about there. We can use pragmatic models very, very, as a problem solving tool, as Starfield kind of advocated long time back, and even look at these complex disease systems, try and find the Achilles heel, make better decisions, make locally relevant decisions. And all of this is possible only if this is like a participatory exercise. A modeler and the public health people and the wildlife biologists all coming together to the table, and they put in the assumptions, make up these rules, design the scenarios, and interpret or analyze the result. So this makes such kind of diseases what the World Health Organization calls tool-ready diseases. We now can have a tool which can give us actionable insights to counter, to mitigate the threats of such diseases, rather than just coming up with a blanket recommendation of rodent control, better precautions, and all of that. So with that, I just want to see if there are any questions. And thank you so much.
So folks, we'll take some Q&A. Since we have a Zoom room, just if you can speak loud enough for Ani to hear you, we're good to go. So we don't need mics. I'm going to start off the questions actually. How do these models, agent-based models, deal with something like SARS-CoV, where a host is going to get infected with one strain and may be transmitting any number of unpredictable strains, not just one strain to one strain, maybe one to many different strains to the population?
Right. We haven't explored that, but what I can say is that is exactly what is needed to bring to the model, because this has immense possibilities of incorporating a multi-strain scenario and a multi-spillover event and all of that. But as I said, because these are individual based models, so what happens is all the individual hosts, you can have multiple species, they have their own set of real world attributes, right? One way of doing what you said is to add the pathogen as an agent. But then that kind of, in my mind, becomes very difficult to have, because you will have millions of those agents, right? And that is not really practical. But what we actually do in there, because these are microparasites, we add the strain as a state variable of the host. And that way we can track it. We can track the changes into like the evolution of that strain. And there are some genotype genetic agent-based modules actually, which can be incorporated, but that is way beyond my skills, and therefore I'm collaborating with the evolutionary person. But that is possible, and that is where we want to go. Thank you. Yes.
I have a question on how you validate those models, because we get resistance on adopting the outcome of models because of lack of validation. So how do you validate those?
That's a great question, and a question that I always kind of, so one thing is for modeling, simulating such complex systems, I would make this like a bold statement here, but there is no way that you could validate the model. What you can do is you can verify the model. What it means is the code is reviewed by people who have expertise in coding, and they can certify that this code does what it says it is doing. So that is code verification, model verification. The other part of the answer to this question, which I frequently get, is validation is required if you want people who are going to use it, if you want them to trust the model, right? I come out and say, don't trust any model, don't trust my models. You parameterize it with your understanding. You kind of like, you can put out a hypothesis saying this is what it should do, and then look if it does that. There is other part to it, which is called pattern oriented modeling, because the patterns emerge out of the model. We don't code for the patterns, right? If those patterns match the real world patterns that you see, then you are kind of in the place where the system that you have modeled, your model is making sense. But to the real answer to your question is, there is no way you can validate such a model because of the stochasticity and uncertainty that goes into it. So if we take out the stochasticity, we could maybe have a model which does exactly what you see in one situation out there. But we want really a general kind of model which should be able to look at different systems. So I don't know if I answered that, but there is no way to validate a model the way you can do simple models, non-complex system model. Yeah. Yes, sir.
Okay,
Please go ahead.
If I want to look into, instead of lepto, where you have 5 to 6 serovirus, or SARS-CoV, maybe you have 10, 12
Yeah.
variants, say Salmonella,
Right.
and you have multiple hosts, whether you have equine or, cattle, and then it's also a food safety pathogen. So the model will get more and more complicated
It will.
because it's multi-host,
It will.
multi-pathogen.
Yeah.
So in those situations, can this modeling be done? Same approach
Right.
locally, or we should look at global trends?
No, that's a great question. I think some people do that kind of modeling. It's very much doable, but it will, as you said, require kind of a different approach. And that is where the modeler has to collaborate with the scientists, with the, maybe the livestock, managers and all of that. That can be done. You will just have to break it into kind of digestible bits, right? Because multiple hosts is possible, multiple, serovar strains is possible, but we still need to ask the right question. Because as you keep on adding state variables to your model or global variables to your model, it becomes heavy. Even though we have made a lot of progress with computational capacity, it still becomes intractable. And so, there are some ways of doing that, but we kind of need to ask the right questions and develop the models as a problem solving tool. We will never be able to mimic the real world complex system, never. There will always be uncertainties. And once we acknowledge that, we can then, if the goal is to find the Achilles heel, then we can just model the necessary components and then try and get the answer. But yeah, yes.
Very interesting talk. I was curious about the SARS-CoV-2 deer model, and because one of the things that's always, I guess, baffled me looking at this white-tailed deer situation is how it gets to deer.
Right.
But do you model like humans at all in that system,
No.
or like points where humans?
So
and
we
deer
have
might
humans in the
connect?
leptospira model as agents, but not in the SARS-CoV-2 model that we have right now. We are just, we kind of have an interface there where in the human circulation can be kind of there is the wastewater surveillance for the region tells us the probability of spillover.
So it's
from-
more like temporal rather than
Yes, but
spatially where it happens.
exactly, yeah, but we can have human agents, but unless and until we have a clear role for why do we want the human agents in there, it can be done. And it might be important for, let's say, park management or tourist kind of place. And so it could be adapted. But I think there are a lot of possibilities. And just making this, I would just finally add to this because this is a very important point. And this might be overstepping, might seem like a very big overarching thing. But I think we should all be working on immortal models. So nobody should have to build all of this again for any other system. Most of this can be used in other model systems. And that is what open science is all about. That is what reproducible science is all about. So like R, right? In R, people write their functions, write their packages and make them available, right? For not only agent-based modeling, but specifically for agent-based modeling, we should have these modules and these model codes accessible, available, so that if you want to put together a human deer interface model, you shouldn't have to go through all the deer biology that a team of 20 biologists did for 10 years. It should be available, it should be used, that reduces the chances of errors, right? And recently there was a paper out by Volker Grimm and others where they exactly talk about this thing, that you can build chunks that are reusable. So, yeah.
So we just have a question
Yeah.
on Zoom, and I'll summarize
Sure.
here. It's from Dr. Kent Reed, and it's about climate.
Okay.
So how do these, can you comment on how these models deal with regional variabilities in climate? And then sort of a follow-up to that is our Minnesota winter
Mhm.
this year was a very sporadic case of non-winter. So
Right.
are those things sort of, I
Yes.
guess, be able to be built
Yes,
in?
Yes, oh, that's a great question. And again, I don't have those components in the two models that I presented here, but this NetLogo kind of system lets us import time series data. Right, so if you have the climate data as a time series for 50 years, the model can go to a particular year and get it in there and then change the conditions and everything accordingly. We just come up with rules, right? So in such a winter, ticks are going to be out early. That is a rule we can very easily put into the model, and the time series data gets in there and then it kind of adapts to that. So that is the strength of this agent-based and NetLogo model. It is not a component of the two models you saw, but it's a very low-hanging fruit. Yeah. Yeah.
One last question.
So whenever you're developing a model, does it involve like any back testing to make sure that the model is matching like historical data before you include like any like intervention parameters?
Yes, yes, exactly. And that is exactly what we did with the chronic wasting disease kind of modeling framework. But that goes back to that pattern-oriented modeling. There are some patterns that we definitely want to emerge out of the model, and they may be historical data or that what is being currently seen. And what conditions result into those patterns is a very important modeling component. And those conditions actually help us better understand the real-world system. And I'll just give you one brief example from another modeling project. There is one disease system called the monkey fever, the Casnur forest disease, an emerging hemorrhagic disease in one part of India, the Western Ghats. And not much is known about it. It's a tick-borne pathogen. One crucial question about that was, is there trans-ovarian transmission in the ticks? Like we have other systems here where we have trans-ovarian transmission. So we use the modeling approach to run two scenarios, one without trans-ovarian transmission and one with, and then looked at the patterns and matched it with the real world patterns to come at a better understanding, like does this play an important role, right? And that is one way to use these models to better understand the dynamics. And it's not, again, a very important thing. These are not predictive, no matter what you do, no matter what anyone says. No model is going to be a crystal ball to predict the future. It will always come with like a caveat that if these conditions are met, this is what you will see. Like COVID vaccination, they said like if you do masking and everything, universities will have only 500 cases. And then first week they had like 2,000 cases because no compliance. So a model can incorporate no compliance, but you have to be very specific. Otherwise, people are going to interpret, as you said, there are going to be 500 cases and we have 5,000. So that's the case. Yeah.
Thank you.
So, Ani, it was fantastic
Thank you.
learning about your research.
Thank you.
Fantastic to see all the collaborations, the rich history that you have with the University of Minnesota, connections that you have here. So on behalf of the CVM, on behalf of the university, we're so happy to keep that tradition alive.
Thank
We
you.
do have some gifts, small gifts for Ani here. Intro hat.
Thank you. Wow.
And Ani has seen the Slinky presentation.
Right.
In our outreach from Impro, we use Slinkies to model how prions become infectious by misfolding. And so, Ani, your very own Impro.
Thank you. Thank you. Thank you.
Okay, folks, we'll just wrap it up here. If you'll join us next month, May 8th, we're gonna have two internal speakers, Dr. Steve Friedenberg and Dr. Noel Noyes, presenting their current research. So please join us on May 8th. And if you could also fill out a survey, which is on the,
We'll get it up there in a second.
which is definitely on the OV or the research office website. And you'll also see it in the emails that we send out advertising the talks. Just a very short 1 to 2 question survey to give us some feedback about how we're doing with the seminar series. So thank you all for joining us. Thank you again,
Thank you.
audience.